Its presentation we appreciate, and I'm sure that all of us learned a lot, and certainly a point for reflection. So our next speaker is Dr. Nima Yazdanpanah. Nima is consultant on advanced manufacturing and modeling and simulation in pharmaceutical and fine chemical industries. His areas of expertise cover modeling and simulation and digitalization process, design and scale up. He has over 20 years of diverse experience in different industry. He has worked in R&D and process design, regulatory CMC and MSAT section at Procegence, where is his company. He supports industry on process development, scale up, tech transfer optimization. His key goals are to apply advanced manufacturing technology and utilize modeling and simulation tools to reduce cost. So that's the highlight. Definitely the industry can benefit from cost saving and getting the products faster to market with proper simulations that Nima is going to talk about. Without further ado, I give you Nima on the stage. Thank you.
Thank you, Firuz. I appreciate it. Good morning, everyone. Do you mind if I stay down there? I prefer to walk, so I cannot stand the seat. Thanks, Dr. Hossain. So you set the stage very well for me about using the models and the science and the knowledge. Let me start with a question. What is QBD? What does it mean? Like how do you make the quality by design, right? Exactly. So based on the ICHQ 87 is knowledge-based process and product development. So the knowledge of knowing the connectivity of different things. What is the impact of your CMA on your CQA? If you change your process parameter, what's going to fail? Or you have impurity, or you have the different properties in your crystals or amorphous dispersion. So that is the basis of this kind of knowledge-based process development. And now we are developing models, and I'm going to show you some mechanistic modeling tools. And mechanistic models are based on the first principle analysis, knowing all the thermodynamics, kinetics, transport phenomena behind all of these, from the molecular level all to the equipments, to see why things are going on and how we can control a process and how we can make a very good quality material. Thanks, Bruce, for the introduction. I'll give you a very quick intro to my company. So it's Procegence. It stands for Process Intelligence. We're a consultancy firm based in Washington, D.C., kind of a startup 5 years ago. So I resigned from FDA early 2020. I started my company February 1st. 1 month after that, COVID hit. So the first year was very challenging, but so we grew. I have no financial investor, no partner, and now we have a kind of eight people. So that shows the value of the service that we do. That has been like organic growth. Our clients very happy, pay for the services, and we have a couple of products into the market that help the companies to fast approval and also the fast process development. So very kind of diverse services and the products, so different modalities, different size of the companies, all over the place from Korea to California. Here you can see some examples in like a small molecule, biologics, drug device combinations, and also some non-pharma. So today I'm just focusing on the spray drying, on the first one, but if you have a question on the other areas, happy to answer questions and discuss more. So that's the approach. So the model that we use is like, oops, sorry, wrong button. So here is like a kind of digital sandbox. So we make a digital models on the computers. We try with different parameters, different properties, different material, without costing any money. I mean, you have to pay me to do this, but like not the cost of the material or like the equipments. Then we have to do the validation when we find the best result. That this validation, again, wrong button, sorry. So this kind of validation could be done in a smaller scale, in the lab scale. Then we can use the validated models for a scale up to very large manufacturing equipments. So you are familiar with the spray drying, so I don't need to go through the basic details. There are different sizes, different shapes, different designs, from like bookie, very kind of a small scale in the lab, all the way to the dairy industry that come like 20-meter highs. And GA, they have like the largest equipment for my client, Catalan. They have PSD7 in Chelsea, Massachusetts, so it's very large equipment. And we have different components in the spray dryer, like from the nozzle to the feed tank to the cyclone, that all impact our process and the quality of the material. So here, I could summarize all my talk in this slide. So this is what's happening in the industry right now. So during the development time in the lab, so we are using a bookie with this type of nozzle. Then we make this type of particles, right? Very thin shell, very fluffy, low bulk density. That is for the lab scale. We are going for the clinical one, or like the formulation developments. We're developing all our tablets or capsules and the dissolution and the stability based on this particle, right? Then we move to phase 2, or like the kind of a scale up to pilot plant. This is like a mobile miner with a different type of nozzle. And now we make different type of particles. Same formulation, right? And again, all the clinical data and all the the medical stuff that go through like over 5 years, 7 years, going to the FDA. So now we get the approval. Now we want to go to manufacturing to larger scale. And now I make these type of particles, right? Now you can see the flesh is larger, so we have a thicker shell. So the dissolution will be different. The disintegration of the tablet will be different. The bioavailability will be different. So everything will be different at the manufacturing scale. But all the clinical data and all the formulation development was based on this type of material, right? So most of the companies now, they have to run the larger scale equipment in a very inefficient way to make a crappy material, crappy particles to match this very bad design from the beginning to match the PBPK and the risk profile, right? Which is not good. Right? So anyway, so scaling up from a smaller scale to manufacturing is very challenging. There are different components there that I'm going to discuss at how we do this kind of a scale up and tech transfer very quickly. But that is kind of very big kind of concern I have, like every time that you have some data from your lab or from the pilot plant that is not be applicable to a larger scale manufacturing, and also worst case for the product. So spray drying, as I said, is a very complex system. We have from the continuous feed is a solution that comes from the heater, from the feeder goes to the heater, goes to the tank. So we have the atomization, drying chamber, now the droplets evaporate, shrinkage, component migration, particle microstructure, everything happen inside the chamber, and then we go to the cyclone. So it's a multi-scale from the molecular level. When we're going like for amorphous dispersion, we have HPMC and I have API. So what will the atomization pressure or the type of the nozzle that I'm using is going to impact my amorphous dispersion. So the particle microstructure from the surface of the tablet to the core of the tablet, you will have a different distribution of the concentration of the material. So this is a different profile, right? So it is a multi-physics. We have heat transfer, mass transfer, momentum, everything involved in this in the system, and also we have the fluid, the solid, all of these things. So spray drying become like very kind of complex process from the mathematical point of view. Similar to crystallization. Crystallization is also one of a very challenging area so we do a lot of services there. So all these what we call like Stefan's problem, which are like the moving boundary condition from the mathematical point of view, or from the pharma, you think spray drying or the crystallization is at the end of the line. The material is expensive, and most of the CQA of the material is being defined there. Residual solvent content, particle size, stability, everything is defined here. So very sensitive process. Now for the scale up, as I mentioned, the CMA and the CQA is mostly fixed, comes from the lab scale. That's what we developed during the formulation and the development time. So we are not going to change my formulation. I'm not going to change the API to excipient ratio. And the CQA is for the bioavailability and the QTTP that we don't change that. But what we can change is the equipment. Is it what type of nozzle I'm using, what type, how much, what kind of pressure in the atomizer I'm using, what's the drying gas temperature. These are like CPPs. And so that gives us kind of wide range of freedom to optimize our process or to develop a design space that if I run at this condition, what I can achieve that kind of CQA. So there is some like a kind of downstream kind of corrective step that we call this as like a controller strategy in the CMC submission. If something goes wrong in my spread dryer, I can run my secondary dryer for a slightly longer time to get to the residual solvent content that I want. Which is not efficient, it's not optimum, but that's been kind of common in the industry these days, right? So in terms of the sensitivities, so we have atomization, which is like very important steps in the spray drying. We have that from the atomization, the droplet size distribution going to be changed, the spray angle. We have droplet and gas mixing and the evaporation, which gives us the shrinkage, and how fast we are drying the particles. It's going to impact my component segregation from the surface to the core. So if we want to have like a perfect kind of mixing and keep it in the kind of solid solution, we need to have like a faster drying or a slowing drying. So it depends on molecular weight of the excipient versus API. Or if you have mixed solvent, let's say if you have ethanol and water, or just pure ethanol. Ethanol water, now the API is soluble in ethanol, and the excipient is soluble in the water, but the vapor pressure is different. So during the spray drying, the ethanol evaporates first. So my API comes to the surface first. I end up with more solid on the surface of the particles. Now we have stability issues because most of these API are not supported or protected by the polymer matrix, right? So that's why we control this kind of drying rate inside the chamber to maintain the quality of the material as well. So we have to start from like developing this knowledge. As I mentioned, like this based on the QBD, so putting all the parameters that connected to each other and gives us different CQAs. So this is a fishbone diagram, so we can develop for different properties. In this case, I'm starting with the residual solvent content, but you can develop same thing for particle size distribution or even like the process yield. What is the connectivity between the parameters, right? So if I can list 100 process parameters, then I have to run sensitivity analysis to figure out what are my like 5 or 10 critical process parameters, right? So developing this knowledge of what is connected to what, and if I change my, let's say, nozzle type, what will impact my atomization, which is going to impact my particle size distribution, right? So that's where the modeling and simulation starts, right? So sitting together with the scientists, with the formulation, with the biologists, with the formulations people to think about all these things that connect together. So that's where we start our model. We don't start from math, right? We start from the science of the formulation and the biology, right? So starting from like atomization, I just go very quickly on the different steps. So the first the first approach would be trial and error. We would have different options for orifice and the core, and also the material properties. If I have different HPMC or PVA or the excipient in my solution, now the rheology of the liquid could be shear thinning or the shear thickening, then it's going to have impact on the droplet size generation. So higher temperature, higher pressure, or the higher atomization gas, or is it that's going to also impact the droplet size distribution. So that's why when we develop this kind of full factorial DOE, is a combination of the equipment, the process parameter, and also the material property. We end up with this kind of 600 kind of full factorial combinations, which is not possible to do in the lab, right? Then we could do some study on like what is like the evaporation capacity of the spray dryer, what is the higher solvent content, what is the gas temperature. Then we get to this kind of center region, which is kind of more efficient to do the high fidelity models for that. For example, now we can see we started doing CFD to predict for different combinations. So this is very hard to read. These are the nozzles from the spray system, like SK1627s and 7625. Those combination of the orifice and the core. Then we get this kind of histogram of the droplet size distribution at each location at a time, right? So atomization, you are all familiar with that. Here is my spray, which is for my hair anyway, so I use it for demonstration here. So this is water, right? And now you can see we have, like with this water, with this atomization pressure, I have this kind of wide angle, right? Now I'm kind of opening up the thread, and so what happened now? It's the same material, same pressure, but with a different now. Now? Oops, maybe more. Now we make a jet, right? Is this, right? Instead of having a spray, with the same equipment, same material, now I have a different droplet size distribution, different velocity, different spray angle. In the GEA equipment, it's called like a spacer. Is a thin, how thick is Andy? Is like half an inch, right? A ring of Teflon that we put on the lens. Then the location of the lens in the chamber goes up and down by like half an inch. Then it's going to have significant impact on your droplet size distribution. It's just when I'm releasing my atomized droplets in the gas, right? Just by half an inch, you can change your particle size distribution quite differently. So that is one example from this simulation. You can see we are doing this kind of atomization simulation there and getting the population balance or cost at different vertical and horizontal location. And what's that important? To develop these histograms to find the velocity if my droplets hitting the wall at this location, and is it still gooey, or is it wet, or is it sticky, but I have enough energy to bounce back. When I hit the wall, it's dry and bounces back. Or no, is a slow velocity still sticky, it's gonna hit the wall and stick there, right? Or what happened at the center, here you can see, now we would have coalescence. The droplets that gonna hit each other and coalesce together. So when you get SEM image from your final product, you can see two different agglomerations. There might be just physical particle kind of sticking together. That's the agglomeration from the cyclone. But if you see some like a bridge between the particles, that is happening during this stage, right? So optimization of the atomization and understanding about how this works is very important. So now we go into like a scale dependencies, like there are different scale dependent and independent. In general, for example, the heat and mass transfer, when you think about, let's say, bioprocessing or hydrogenation, right, the KLA, these are all scale dependent phenomena. But reaction kinetic is a scale independent, that's thermodynamic property, right? So that's for a scale up of the spread drying, we have to consider all of these, all of these properties or the aspects that are scale dependent when I'm increasing my chamber size, or when I change the diameter to height ratio of my chamber, so the residence time would be different in the chamber, right? These are all giving us different evaporation rate, different particle shrinkage time. Now you can see here we're comparing the PSD3 and PSD4. These are like normalized height, so based on like the same PSD4. And when we design these evaporation models and put it in the simulation, now we can predict that how much evaporation I have, what is the mass transfer gradient that we have at each stage. And if I want to transfer from PSD3 to PSD4, or I have a site in Europe that has been using PSD3, now I want to come to US with the PSD4, how we can do this kind of overlap of the knowledge and this kind of tech transfer very, very quickly. In this case, you can see PSD4 has a higher capacity, even with the same rate. So when the PSD3 become kind of flat, because it has like a lower evaporation capacity, this is a smaller equipment, right? But at the same rate, so we would have more capacity from the PSD4. That is kind of non-trivial because it's a bigger equipment, but when you compare to the ratio of residence time, PSD3, PSD4 is almost kind of same height, but the diameter is different, so the residence time inside the chamber is different. That is the residence time in the chamber. So this is for the same material, same properties, same thing. We have D10, D50, D90. Now we can see D10 stays longer time in the chamber. D90 is heavier, is larger. It leaves the chamber in a shorter time, right? Now, what's going to be the problem? So small particles, the surface to to bulk ratio is larger. Now we have larger mass transfer flux because the surface area is larger, right? And also we have larger heat transfer, so they are exposing to a higher temperature. Now my D10 stays in a chamber for longer time, which is time, right? And exposed to higher temperature, which is activation energy. So now think about the reaction kinetic. Most of the degradation, most of the impurities happening in the D10. D90, we have a large particles. Now the diffusion length is larger, so it takes longer time these particles to dry, and they stay shorter time in the chamber, right? So most of my residual solvent will be in D90. So we had a client 4 years ago, and they had impurity problem. I told them that remove your D10, physically remove your D10, your chemical purity goes up. And the formulation guys, they said, no, that's BS, it's not gonna happen. So they tested and it worked, right? Because it is a science. You know, I'm exposing the smaller material to longer time, and we're gonna have the more degradation there. Now, especially if you are designing the secondary dryer, it could be double cone, it could be tray, whatever design. Now you have to think about, you know, if you have a bulk, average bulk moisture content, you have to normalize based on each particle beans. My D10 is gonna dry faster, and in a shorter time, my D90 gonna dry, and it takes longer time. So we have to do this kind of breakdown of these particles and the moisture content per size of the material. Cyclone, so that's more for like the design of the cyclone, and now I'm going to get into like also the regulatory submissions, that how we are using the models to support the CMC, and these models could be used for like risk assessment, process development, and also the PAR and NOR and the design space. This example is for cyclone. That was the efficiency of the cyclone, right? You can see most of my D10 kind of escaping the cyclone at this rate, at this flow rate of the gas. Most of the D90 going to be all captured. Now, if you are changing the gas flow rates, my D10 would be less, and you know the particle size distribution is the statistical distribution of the sizes per beans, right? When I'm losing a portion of my D10, the whole thing shifts toward the D90, right? That's the impact of the particle, the drying gas rate, the flow rate of the drying gas on your particle size distribution. Nothing to do with the evaporation and the shrinkage yet, right? Is all in the cyclone. That's one example. We have all the problems in the chamber later on, so I'm gonna get to that as well. So now we get into like parametric studies, running a lot of what-if condition. Now we can think about what if I keep my feed flow rate fixed, drying gas flow rate fixed, but changing the drying gas temperature. What would be the impact on the powder temperature and the solvent content? Now we are running this kind of parametric study for different steps, for different intervals, and we get to some kind of knowledge or like the connectivity of the parameters. Or the next case study, if I keep the feed flow rate changing, what will be the impact of the temperature and the solvent content, and running multiple of these, which is going to be like very cheap, very fast now, because I can run one of these in like 10 minutes, right? This is now we develop the models, now we have the fully high fidelity simulations based on the particles and the droplets and the chambers and everything. Now running this kind of 10 experiment, 20 experiment just takes 1 hour, right? So there's no way you could do the same level of the kind of exploration in the lab or in the manufacturing. Now we are talking about like PST3, which when you work with like a CMOs, how much you CMOs charge per day for PST3, PST4? Nobody going to tell me? No. Is something cost around like $100,000 per day-ish, right? So this would cost like $1 million if you want to run in the lab or in the manufacturing. So now combining many of these case studies together, and we get to this kind of response surface. Now from here, I can use it for 3 different things. That gives me the design space. If I change my temperature where I end up in the flow rate or in material temperature, that goes to the CMC package for regulatory submission. This also could go to the advanced process control for for this kind of dynamic control of the process. If your feed goes down, how I can bring up the temperature to maintain the CQA. You may think that this is very difficult in a regulated industry. We have done that before recently 2 years ago, and I can show you the results and how FDA liked that approach as well. So that's like the design space. If you remember, here is more like kind of theoretical design space. Yes, I can run my spray dryer at 1 million degrees C, but it's not possible in the physical world because it's like, you know, a very hot condition. Now we're putting all these kind of limits on like what is the lowest solvent I can, what's the highest solvent content I reach, what's the highest temperature, yield, economy, all of these, and get to this kind of design space that is safe, that is I can run my equipment at the larger scale manufacturing and gives me this kind of confidence in terms of the quality, yield, and the cost of the process. So I'm running out of the time. So I'll give you one or two examples. Here it was a case study. Actually, the client was here in San Diego, in New Jersey. They had a peptide. It was very expensive material. Wanted to kind of scale up from a pilot to larger scale manufacturing. They didn't know the process parameters or what kind of CMA they have to use, or they have to change their excipient, or how much they have to charge in the feed vessel. So that's the big problem they had, and also they didn't have enough material. Just had like 10 kilo of the peptides. Now running 1 day of a spray dryer needs like 130 kilo of the peptides. What you can do? Right? So they thought about like reconstituting the material, and every time making a feed, then it's not a continuous process. I mean, you have to break down every like 10 minutes, every 10 hours to make the feed again. So, and every time that the rear spray dries, so because of the peptide is heat sensitive, so the impurity goes up and up and up, right? So that's what they came to us. So the CMO asked them for like 12 days DOE, along $1 million cost, and so we helped them with this developing the models. They had a very fast process development, product development, and they saved $800,000 in like 4 days. You may say, okay, that's good story, but how about the validation, right? So all these models are sexy, colorful, and I can run a lot of smoke and mirrors and fake things, right? But when we go to the actual process, we need to also demonstrate the validation. Here's an example. So Esteve Chemical in Barcelona. So that this example come from them. That's our client, and they said, okay, we don't believe in math. We believe on what we see. Show your simulation results first, then we compare with our experimental data, right? So it was a kind of blind test. And now you can see for solvent content, temperature, and the particle size distribution, and that was a very complex formulation in terms of the solvent composition and the material. So we had API, excipient, and a surfactant there, and a binder. And you can see for dichloromethane and the methanol are error. So the blue one is error. The greens are modern results, the purples are experimental data, right? So we had less than 5% error. For temperature, we had like outlet temperature 0% error, 4% error for the inlet temperature, and particle size distribution, D10, 2% error, 6% error, and 9% error, right? So these are like validated with the experimental data with a third party. So everyone was so happy. They said, oh, we didn't even expect 30% error, so this 10% is marvelous, is fascinating. When I said, absolutely, I have a problem here at D90. Why our D90 error is too high? And they say, you're stupid. We are happy with the results. Why you want to change this? So now we have started running different tests. How they measure the particle size distribution is by like a laser sheets, right? So you scoop some powders, you put through this laser diffraction, then you get like a statistical average of the particles, right? But what we are simulating is like individual droplets, individual particles. Now we get to this problem. Now we collected a couple of SEM images and run the image analysis for individual droplets. You see we are spot on. So like error was like 2%, 3%. But now we have these kind of agglomerates, these are not like large particles. They're like the clumps of small particles together that could happen on the cyclone wall, on the surface of everything. And now these large things that would go through my laser diffraction and skew up the entire particle size distribution to D19, right? So if you If you have your powders from a spray dryer, just shake them or put them on like a vibration ultrasound, your D90 goes down, right? Because you're kind of breaking down these are not interconnected, it's just sitting to each other. If you separate them, your D90 would go down, right? So anyway, so that was kind of a nice study that particle size was measured differently. So we need to have this kind of like the orthogonal test. So we did the laser diffraction. I mean, they did it first, then we had to run for the SEM images, and we had this kind of nice validation between the experimental data and the simulations. So I have many more backup slides. Please ask me more questions that I would have a chance to show them. So as a quick summary, is again going back to my first question, what is the QVD? You need to understand what the connectivity of different things. What is the CMA? They're connected to my quality attributes. If I change my process parameter, what's going to impact my material, my process, my product, my everything, stability, the patients, tablets. In terms of the process development time and the cost, these models are very kind of time saving and the cost saving for during the development time. Also, when we have optimized process, it's going to be continuous saving over time, right? So instead of costing, let's say, $1 per kilo of the material in this kind of bad design, now I'm making 50 cents per kilo. It's going to be continuously, I'm saving 50% over the next 20 years. Right? That's going to be a lot of kind of time saving. So we had a client, I don't want to name them in the recorded session. So we helped them to bring the product to market 9 months faster. Right? During the process development, during the submissions, we also submitted the models to FDA, and FDA loved that the model results, and they had very fast review. When you have a blockbuster, the value of 1 day drug in the market is like $1 million per day. Right? So 9 months is $300 million saving. They didn't pay me that much, but yeah, so that's the saving they had. Thanks for the time, thanks for your attention. I hope I could be showing you some cool stuff, and if you have any question, happy to answer.
Thank you, Nima. Any quick questions before we move on to our next speaker for this morning break? Yes.
Nima, actually,
He's off.
I'm a drug substance guy.
Huh?
Yeah. So I'm a drug substance guy. So we saw each other.
Yes, yes, thank you.
But anyway, the downscaling for scaling up is very common in drug substance area.
Yes, yeah,
So
yeah.
can you tell me about your experience in terms of downscaling for scaling up?
Yes, I have backup slides. I'm not sure how we can manage this. I don't know where they are then. Never mind. That's fine. Don't worry. So scaling down in a spread, for example, as I mentioned, like hydrogenation, right? So is it for drug substance? You see it is.
Especially for the drug product, like
Oh, I see. A spray drying. So no, it's not going to work. Because when you scale down, you would have a different nozzle. Could you go to slide 2, please? Sorry, don't want to click back. I cannot count, sorry, this one. When you scale down, so this is your final goal, right? So this is the high pressure atomizer that you can use in this spray dryer. If you want to go scale down, you cannot use high pressure atomizer in something like Buki. You can buy one of the Saran's equipment, but that will like a different story, right? Or like the Procept. So scale down now, you have to change the rheology of your material. So you have to go to lower HPMC or PVA, because if you go with a very thick viscosity, it cannot go through this nozzle. Right? So the scale down in this case is not going to work, because it's not the same equipment anymore. It's going to be totally different new equipment.
Did you simulate the bigger particle size with the ultrasonic nozzle? nozzle or something else rather than pressure nozzle?
Yes, yeah, you can make like, as you mentioned, like this membrane or piezoelectrics. There are different ways to make the different particles, right? What's happening here, like at what we call it like the interfacial forces. When we are releasing one droplets from this core, so how much pressure I'm putting on this skin, right? So if I kind of gently touch my skin, there's nothing happen, right? But if I kind of apply too much pressure, you can see it become red, right? It's similar to droplets. So when we are going through this high pressure nozzle or the orifice, because you have this the gun of the gas, right? So that kind of interfacial forces cause also some kind of like internal mixing in the droplets. It's some sort of like a Marangoni effect, right? That also could cause some like convective separation of your low molecular weight, like the API, and the heavy material, large molecular weight, they go on the center. So it's going to be totally different. Yeah. There was a question there, I think, if you have time.
Yeah, we got.
You can go.
Oh, you're fine. Without giving away too much, thank you very much for the talk. What would you say the greatest source of error in your models is?
Right. Shrinkage rate and diffusion rate. Right. So for these cases, we ask our client to run DVS, dynamic vapor absorption, with the solvent that is being used in a spray dryer. But we need to get a desorption curve, not the absorption curve. They're going to be a hysteresis, right? So the absorption go here, the desorption comes back. And when we know the length of the material, the diffusion length or the particle size, right? Now from the DVS, you get the mass change per time. Per time gives me the rate. Now I have the mass change times the diffusion rate, the diffusion length, given the diffusivity. That is kind of a very high error area that there. Yeah. Let's go. Thanks.
Thank you.
Thank you.
Thank you, Nima.